Hi, thanks for joining. My name is Aitor, and I, together with Rembrandt, will be presenting us things. We will be presenting our 3D reconstruction of the macaque brain. We are both, in case you don't know us, from Jülich, working at the INM6, just across the street from where Timo and Katrin have their offices. So what we want to do is, Rembrandt, you can skip, yeah, thanks. Is to actually build a model. So we are from the modeling side. However, our models include lots of anatomical details, such as the number of neurons and the connectivity and so on. And what we require would be a very accurate cell counts for all of the areas across the macaque cortex. And this do unfortunately not exist for every area, and also especially not exist in a layer resolved fashion, which is then what we are trying to obtain with this reconstruction. So for this, we needed to find some histological data, which Rembrandt will be telling you about. And then, of course, we have to stack these images in a reasonable way in 3 dimensions, which we then register to an atlas. And this atlas we chose to be M132 Atlas, which was presented with Markov et al. In 2013. And the reason for this being that the connectivity data that they measured by tracing in these studies is in this atlas, and also the previous cell counts and cortical thicknesses and so on also have been measured in this atlas. So it just makes sense for us to keep on with this one.
So then when looking for histological data, we found two public resources on the, basically on the web. The first one is the NIH Blueprint Non-Human Primate data set, which is measured about 12 years ago or even more. So it has been sitting there, and it seems that not so much has happened to it. So there is no volume already constructed from it, to our big surprise. It has also different ages of monkeys. We took the adult, and it has a lot of gene expression data, which we did not consider. So the resolution of that data is like 1 micrometer in plane and 50 micrometers slice thickness, where they discarded 4 other sections. So the slice spacing is 250 microns. And there's also another data set that covers the complete brain, which is now hosted by Roberto Toro and Katja Hoyer, who are also among the audience. And this is provided by Edward Jones. And we may look into that later if we can get the time to do that. So to create a volume out of this histological data, we need a reference space to guide this process. And for that, we chose the high resolution MRI that comes with the Calabrese et al. Atlas from 2015. This is actually an averaged MRI, so it's a very nice, smooth, high-resolution volume.
Okay, so yeah, basically, just to start with the process, we took these slices and we downscaled them to a more manageable size, and also took just a grayscale of them. We also drew some outlines of all the cortex, including all of the sulci and so on, because these then are very important landmarks in the registration process. And we masked our slices with that, so removing background essentially. And then we smoothened, and we also equalized the gray values of our images, so basically to have a uniform color across the entire volume. And then build one initial volume, which would look something like this. So it's very, very rough and very non-smooth. Which was made by just aligning across the centers of mass.
Okay, have another look at figure D. We are going to call this data set the hedgehog data set. Okay, now we have our initial volume, and we want to construct a smooth, well-behaved volume out of that. So there is a toolbox published by Piotr Majka in 2016. It's called poSSum. And this is the one that we were going to use. Well, to get it started, there was actually a lot of black smoke coming from the exhaust, and the batteries were also drained. So we spent a lot of time of making it up to date with Python 3, which we released on the GitHub repository. And then we applied some of its algorithms. The first step is a very important one. It's iterative pairwise alignment. What you do is you have a reference volume, the MRI, you register it to the initial raw volume, which is figure D. And then for each experimental section, you have a corresponding MRI section. And then you start to do 2D to 2D registration of all these sections to the MRI section. And the volume gets much smoother. And then you repeat this process whereby the MRI may rotate a little bit. So you reslice it every time. And then you go from D to E, which is an important step. Then the next algorithm that we tried was sequential alignment, where you use the fact that adjacent sections, or sometimes the second next section, have very similar contrasts. So if you register them to each other, you also get a smooth volume out of the sections. It just suffers from one side effect, is that like low frequency parts in the data are not captured well, and this is called the banana effect. So actually the result of this algorithm was the yellow figure. So the shape of the brain got changed a lot. But there's an easy way to undo that. You can take a look at affine transformations and take out the low frequency part. So then you get back to the gray picture again. There's not a big improvement between E and F. So this step may even be skipped. We have not tried that, but it's not such a big improvement. And then we do the final step, where you do basically the same as you do from E to F, but now with deformable transformations. But if you would just blindly do that, you would again have the banana effect, or maybe even worse, because you have more degrees of freedom. And there is not easy way to undo it if you do deformable transformations. So instead, we used an extra cost term in the cost function, where we also tried to align the sections to the Calabrese MRI sections as we did it in the step from D to E. So it's more or less the two steps combined to go to G. And now I'm going to show you the final result of this procedure, at least in three planes. So the coronal plane is the native slice direction, slicing plane. So there you see, of course, a very good match. Then if you look at the sagittal plane and the horizontal planes, well, there are some places where there are just some artifacts in the section, and I'm showing you an example here. There's some missing data in this slice, but in general, the slices are very clean and well behaved. So there's not too much to repair if you want to make this more or less perfect. And here you can see the 3D reconstruction of the histological volume.
Right. So we were quite happy with our reconstruction so far. I mean, there's still some refinement to the steps that can be done. And then the other major step we needed to take in order to find like quantitative measurements is to get the atlas also in the same space. So what we did is we did a nonlinear registration, which is very standard, and we also added some corresponding points to it to improve it, so to force the sulci and so on to be aligned to each other in the two spaces. And this produced this image you see here in the middle. And then we also came up with some post-processing steps, which cause a few problems, but in general, they work also quite well, where the main idea is to fill out all of the cortex with some label, because we want all the cortex to have some label at least. And then we need still to do some refinement to our post-processing, especially because some areas tend to jump across sulcus to the other side. And so this is, then really a problem if we want to measure things like volumes or other kinds of measurements. And once we have our atlas in the exact same space as the MRI, and also pretty much the same space as where we registered our volume, we can undo this and we can apply all the steps that Rembrandt just told you about backwards to get our atlas on top of our original histological slices, which then have no deformation of the neurons, and where we can then count neurons. So if you move to the next slide, yeah, we come up with something like this here on the top, where we have essentially an overlay of the atlas on top of every single slice, as it was in the original slices. And then we can already start, I mean, obviously with taking care that the registration is not perfect, to extract some samples where we can then count cells, because this is also 1 micrometer resolution. I don't know if this was said. So we can then, yeah, exactly, start counting cells, and this is as far as we have come.
So then we jump to conclusions. So in the process of discounting cells, we came across some histological data set, which we constructed a 3D volume out of. And this, of course, reminded us of the BigBrain, which is this wonderful isotropic volume of data. So such a data set is not yet available for the macaque. So maybe this non-isotropic volume with a rather thick slice dimension can still serve as a reference volume until better data gets measured for the macaque. So the idea is to use this in the eBrains and BigBrain machinery to expose this volume to people who want to do some analysis to it, because we're also working in the same time on a new non-human primate atlas to contribute to eBrains, and this could be a nice joint project. So we needed to choose some in-between media, the Calabrese high-resolution MRI we used, because you cannot easily map directly just a cortical parcellation into a set of sections. But we might as well take the upcoming NHP atlas as an intermediary. So we would like to hear from the BigBrain team if we can improve on our pipeline, because we are kind of beginners in this field. And I'm sure there is a lot of tips that you can give to us. Thank you very much.
Great, thank you very much for a brilliant talk.